Hello, and thank you for watching my talk. The title of my talk is Hydroxyl Radical Measurements of Simulated Lunar Dust in Biological Fluids. The purpose of my work is to understand the health effects of lunar dust, and I do that by assessing the reactivity or the hydroxyl radical generation of minerals in lunar simulants and try to connect that to potential health risks. And we look at hydroxyl radical generation because OH radical is known to be cancerous to lung tissues. So I am part of the RISE 2.0 team, and within that team, I am part of the Theme 3 group. And the work of that group is to probe the health effects of lunar dust. And so we do that in multiple ways. One, by developing the next generation of lunar simulants. Two, assessing the reactivity of these simulants in solution, which is what I'm going to talk about today. And three, by evaluating the toxicity of these simulants in cells and in live animals. And so far what we've done is we've obtained 3 different lunar simulants and reduced them to simulate some of the aspects of space weathering. And we've done this with JSC1A, LMS1, and LHS1. We have measured the hydroxyl radical generated by these materials, incubated in biological fluids using EPR spectroscopy. And we've done this in both simulated lung fluid and in artificial lysosomal fluid. And so LMS1 is Lunar Mare Simulant 1, and LHS1 is Lunar Highland Simulant 1. And these simulants are better than JSC1A because the mineral proportions are more in line with what you find in the Lunar Mare and the Lunar Highlands. But these do also have their drawbacks similar to JSC1A in that they are deficient in iron and calcium and that they have excessive magnesium, sodium, and potassium. But one thing that all of these simulants have in common is that there is no agglutinated glass, nor is there any metallic iron. So here in this list, we have the components present in both the simulated lung fluid and artificial lysosomal fluid in grams per liter. The green represents the amino acids present in both of the fluids. And in red, we have DPPC, which is a surfactant that is commonly found in lung fluid. And so simulated lung fluid is the fluid that coats the passageways of the lungs. So we have numerous passageways throughout our lungs, and that's all coated in a fluid, and the simulated lung fluid is supposed to represent that. The artificial lysosomal fluid represents the inside environment of macrophages. And so when you inhale a particle, macrophages will try and grab these particles through a process known as phagocytosis. And so when this happens, those particles are then able to be removed by those macrophages. And so that's what ALF is trying to represent, the environment of those macrophages. So in order to make our lunar simulants more lunar-like, we added metallic iron to them. And so we did that by reducing them in a stream of hydrogen gas at 900 degrees Celsius. So what we observe is that upon removing the sample, each of the lunar simulants gets a little bit darker, indicating the likelihood that we did add metallic iron to the surface. This is an SEM image of a few JSC1A grains, and all these blue arrows are pointing to different blebs on the surfaces of the grains. And these blebs are very likely metallic iron. There was another paper by Allen et al. In 1994 that did a similar experiment with a few mineral grains, and they found that the similar procedure, the heat treatment and the reduction, produced these blebs, and they were able to use mass spectroscopy to determine that they were in fact metallic iron particles. And so to measure the amount of hydroxyl radical generated by each of the lunar simulants, what we did was we used a technique called electron paramagnetic resonance spectroscopy, or EPR spectroscopy. What we did was we used a compound called DMPO, circled here in the red, which then traps any of the hydroxyl radical generated by the simulant, which is here circled in green, to form this stable compound, radical compound, circled here in blue. So what we did was we measured each lunar simulant at a particle loading of 100 meters squared per liter. So that was incubated in both the biological fluid and the DMPO solution. And so that was incubated for 15 minutes. It was then filtered using a syringe filter, and then we inserted the filtrate into a capillary tube, which is then measured with EPR. And so these are the results using just DI water. So we substituted DI water instead of the ALF and the SLF. And so here we can see very stark differences. So the bars in gray represent the non-reduced and heat-treated samples, and the bars in red represent the reduced and heat-treated samples. And as you can see, there is a massive change in the reactivity, or the amount of hydroxyl radical generated in solution by reduced samples versus those that are not. And so the difference, or about the average difference in between the reduced and non-reduced materials, is about 10 to 20 times. So this is a huge change in reactivity just upon the heat treatment and the reduction by themselves. And this increase in reactivity is very likely due to the formation of the metallic iron that was formed on the surfaces of all of these lunar simulants. And there was a paper, Wallace et al. 2010, which compared the reactivity of Apollo samples with JSC1A, with non-reduced JSC1A. And it was found that the reactivity of the lunar samples, the Apollo samples, were much more reactive compared to the JSC1A. And they also attributed this to the presence of metallic iron. They were able to see a strong correlation between maturity, so the amount of iron compared to total iron, and the ability to generate hydroxyl radical in solution. So the difference in reactivity between our reduced and non-reduced samples is actually higher compared to what they saw in Wallace et al. 2010. And that is likely due to the fact that those Apollo samples may not have been as pristine because they were not well kept in the curation facility for 40 years. And our samples are freshly reduced, so it is likely that since our samples are freshly made, that these in fact may possibly be more representative of the reactivity of fresh lunar samples, although of course to prove that we would need a lot more study. So as you can see here, the results in the artificial lysosomal fluid and in the simulated lung fluid are quite different. There is virtually no detectable hydroxyl radical that was measured in either of the solutions for any of the samples, even for the reduced samples. And this could be for a multitude of reasons, and this does not necessarily indicate that there is no OH radical being made. So there's many possibilities and many things that could be happening here. So one possibility is that some of the electrolytes and the other components present in both of these fluids could be preventing any surface iron, whether it be reduced iron or ferrous iron, on the surfaces of these simulants from generating hydroxyl radicals, or it could be preventing other surface sites on these simulants from participating in any type of chemistry that would generate hydroxyl radicals. So that's one possibility. So it could be that these do in fact suppress hydroxyl radicals. It is also possible that components like cysteine and glycine, which are amino acids, could be consuming the hydroxyl radical and doing so at a rate faster than DMPO, the spin trap compound, can trap OH radical in order to measure the amount of OH radical being made. So keep in mind that we're not measuring OH radical directly, we're measuring the amount of the DMPO and OH being formed together. So the DMPO reacts with the OH radical, which then forms a DMPO-OH adduct that is then measured. And that's what we measure. So if the rate of reaction of the hydroxyl radical with the amino acids is faster than that of the DMPO, then it could be that we're measuring a lot of OH radical, or that there is a lot of OH radical. It's just that we're not able to measure it because the DMPO-OH radical reaction is slower. So that's also another possibility as well. It was expected that the reactivity in ALF would actually be significantly higher than SLF and even DI water, and that's because the pH of ALF is 4.5, so that's a more aggressive solution. And so it was expected that the amount of OH radical measured, of course, would be higher, and that was not the case. Something we found when we incubated the simulants in the artificial lysosomal fluid was that the filtrate was not clear. It was a pale yellow color, indicating that one of the components of ALF was able to leach away different surface sites, possibly iron, from the lunar simulants and generate other compounds and interfere with the measurement of the OH radical. And so we cannot say that there is no reactivity present or no OH radical being generated in these fluids. But what we can likely say is that there is a suppression of OH radical and a consumption of OH radical by some of the components in these two fluids. So let's recap some of the biofluid reactivity. And so OH radical generation rises sharply in DI water for the heat-treated simulants versus those that were not heat-treated, and this is likely due to the presence of metallic iron. So there was no measurable signal of reactivity in the SLF or the ALF, and this is likely due to a multitude of reasons. First being that iron on the surfaces of these grains are chelated, which limits the generation of hydroxyl radical. And there's other components on the surfaces of these grains that may also contribute to the generation of OH radical, which are also chelated by these components. It is also very likely that the amino acids, particularly in SLF, since it has more amino acids compared to ALF, are consumed by the OH radical generated at a rate faster than they can be trapped by the spin trap compound. And so this has many negative health implications because any inhaled lunar dust could react with proteins present in lung fluid, which can then cause these proteins to denature. ALF includes components such as sodium tartrate and high levels of sodium hydroxide and citric acid, so it may be that any of the surface sites on these dust grains that are responsible for generating hydroxyl radicals are being dissolved, and so you're not getting anything. So to conclude, heat treatment and reduction of lunar simulants to make them more lunar-like leads to a massive increase in the measured hydroxyl radical generation. There was no measured hydroxyl radicals in SLF and ALF, likely due to the inhibition of some of the surface sites of some of these simulants by the components in the biofluids. Or it could be that the hydroxyl radicals were consumed by the amino acids in the biofluids, and in all likelihood, it might be both. So what we started to do is to measure the generation of hydroxyl radicals in the individual components themselves. So SLF has, I think, like 9 different components, and so what if we test the reactivity instead of all the 9 components combined, each individual one, to see how the reactivity is affected, and maybe that some components don't affect it at all. Some may increase the reactivity, some may decrease it. And so we started this with San Carlos olivine. So we have plenty of San Carlos olivine, so we decided to kind of do this sort of as a test. And we found that the generation of OH radical in the amino acids of SLF, so just the cysteine and the glycine, is actually lower than in the amino acid component of ALF. So ALF has a low concentration of glycine, in the concentration of glycine and cysteine, and SLF is much higher. And so we found that the generation of OH radical in the SLF amino acids is actually lower. So we have some evidence indicating that the amino acids may be playing a huge factor in the observed decrease in reactivity compared to the reactivity measurements in DI water. And I'd like to thank all the people in this list for all their help and support. And I look forward to your questions during the conference. Thank you.